DNA transcription and translation. So this video will cover from the actual code in DNA all the way to actually making the final protein. Because in the end of the day, the entire point of DNA is to encode for proteins. As we should already know, genes are coded DNA instructions that control the production of proteins that cause the phenotypes that you saw in genetics. It used to be thought that most of our DNA was genes that actually encoded for proteins. However, it's been discovered that the vast majority of your DNA does not code for proteins, and it was called junk DNA. But it's been even more recently discovered that this junk DNA is not actually junk. It has a lot of other regulatory control mechanisms and is extremely important in expressing different genes. So how do you actually make a protein from DNA? Well, the first step is you have to first copy the sequence of DNA and change it into an RNA sequence, an RNA strand. So DNA is the actual book that gives the instructions, while RNA is going to copy that, and we're going to use that to actually build the protein. So RNA actually contains the coded information for making the protein. DNA is your encyclopedia. RNA is going to copy down specific parts of that encyclopedia and use that to then make the protein. So we've already discussed RNA structure, but there's 4 differences that you should already know. One is that the sugar in RNA is ribose instead of deoxyribose, which is why it's RNA and not DNA. RNA is usually single-stranded, while DNA is usually double-stranded. RNA has uracil instead of thymine as one of the nitrogen bases. And DNA will always stay inside the nucleus, while the RNA can actually leave the nucleus and move to the cytoplasm during translation. There are 3 types of RNA that you're responsible for knowing. First one is known as messenger RNA, which stands for mRNA. We'll go over each one separately in a second. Second one is known as ribosomal RNA, or rRNA, which I hope you can tell is clearly part of the ribosome, since it's called a ribosomal. And the last one is known as transfer RNA, or tRNA. The first type of RNA is known as messenger RNA, or mRNA. It's called messenger RNA because it's actually going to carry the copy of the instructions from the DNA for assembling the amino acids into proteins. So it's carrying the message of how to actually make the protein from the DNA in the nucleus to where the ribosomes are located. Second type, ribosomal RNA. Ribosomes themselves, you see the picture at the top, are organelles comprised both of proteins and ribosomal RNA, or rRNA. So that's their main function, is that they're part of the ribosome. And the last type of RNA is known as transfer RNA, or tRNA. The reason why it's called transfer, because it's going to transfer the amino acid floating around in the cytoplasm to the actual site of protein synthesis. So it's going to move the amino acid from the floating around the cytoplasm to the ribosome, where the amino acids are being linked into protein chains, or polypeptides. The first main part of protein synthesis is known as transcription. So it's the first step in making proteins. It's the process of taking one gene, DNA, and converting it into a messenger RNA strand. So you're going from DNA to RNA. The reason why it's called transcription is when you transcribe something, you're copying it. So if I asked you to transcribe the dictionary, you'd have to copy every word in the dictionary and every definition. So you're copying the message from DNA and creating a messenger RNA piece with that same information. And it occurs in the nucleus, since that's where the DNA is located. So you are responsible for knowing where transcription actually occurs. This is a diagram showing transcription occurring. So you can see there's actually 3 main phases of transcription: initiation, elongation, and termination. We're not going into each of them in that much detail. The next few slides will go over the steps that I expect you to know. One thing I would like to point out, though, in the first picture, in the initiation, you notice there's 2 parts of the DNA molecule that are labeled, the promoter and the terminator. The promoter region is important for starting transcription, while the terminator region is important for stopping it and telling the RNA polymerase when to release and stop making messenger RNA. And you can see at the very bottom it shows you over time how the messenger RNA, the light green strand, is being made and is slowly being released from the DNA duplex. What are the actual steps to transcription that I expect you to know? Step 1, an enzyme attaches to the promoter region, which is the start signal region of a gene, and actually unwinds the DNA. So it's opening it up, allowing other enzymes to have access to it. Step 2, one of the DNA strands acts as a template, so it is known as the template strand. So you can see in the picture, you have the initiation site by the promoter. The DNA is being unwound, and this new enzyme called RNA polymerase is attaching to the DNA and is actually making the messenger RNA. Third step, the mRNA copy is made from the DNA template by an enzyme known as RNA polymerase. So DNA polymerase makes DNA, RNA polymerase makes RNA. And also notice that RNA is made in the same direction as DNA was. RNA must be made in the 5' to 3' direction. There's only one, that is the only direction that RNA and DNA can be made in. The mRNA copy is made until the RNA polymerase reaches the termination or the stop sequence. At that point, the RNA will release itself, and the messenger RNA is complete and ready to move to the cytoplasm. And then finally, the two strands of DNA then rejoin. This shows you a blown up picture of the RNA polymerase creating the messenger RNA molecule. You notice that the RNA nucleotides are added to the 3' end, just like in DNA synthesis, so it's 5' to 3'. You notice that the RNA nucleotides are complementary to the DNA strand, so they do undergo base pairing, normal base pairing. And you see that the transcription bubble is moving to the right. But that's all I want you to see of this. Just want you to notice that the nucleotides are added to the 3' end, and they are complementary to the actual template strand of the DNA. So let's do an example together, transcribing this DNA molecule into messenger RNA. So you see that this is the template strand of the DNA, and our job is to create the complementary messenger RNA strand. So let's say that this is your 5' end, this is your 3' end. Messenger RNA is always made in the 5' to 3' direction. Therefore, the messenger RNA, this side will be 5', and this side will be 3'. So remember, in RNA, the base pairings are adenine base pairs to uracil, since RNA does not have thymine and has uracil instead, and cytosine base pairs to guanine. So now we just fill in the messenger RNA using these base pairing rules. So we have C, G, G, U, A, U, G, C, U, A, G, C, U, and A. So you can see the bottom strand is our messenger RNA, which will then move to the cytoplasm. So remember, the entire point of making messenger RNA is to eventually take that message and create the finished protein. Without proteins, we wouldn't survive. As we already know, proteins are made of amino acids, so proteins are polymers of amino acids. Amino acids linked together in big long chains. We already talked about there only being 20 different amino acids out there. You do not need to know the name of the 20, you just need to know there are 20 amino acids. But if there's only 20 amino acids, but hundreds of thousands of different proteins in your body alone. So the question is, how could 20 amino acids create hundreds of thousands of different proteins? So let's compare it to something that we do understand, hopefully, the English language. So how many letters are in the alphabet? A, B, C, D. We all should know there's 26 letters in the alphabet, English alphabet. But how many words are there in English? Miss, ings, is, smart. There's also almost an infinite amount of words. So each word just has a unique structure of letters, just like each protein has a unique structure of amino acids. So just how Miss and Smart are different, two different proteins can just have the same letters or same amino acids, just in a different order, or the amount might be different. We've also already mentioned this before. The actual bonds that link the amino acids together are known as peptide bonds. Peptide for protein. So you can see here the peptide bond forms between the amino side of one amino acid and the carboxyl side of the other amino acid. And water is actually released during the formation. I'm not going to go into a lot of detail with protein structure, but you can see originally at the top you start out with a strain of amino acids. Those amino acids will then interact with each other to form a secondary structure, where you can see it forms a different shape. Those different shapes then can interact with other amino acid chains, other polypeptides, to form the final protein at the bottom. So you can see it's amazing how it just goes from a strand of different amino acids to eventually an actual three-dimensional structure that is extremely important in the function of this protein. We have successfully copied the message from DNA. Now we have to translate that message to actually build our protein. So that's called translation. So it's the production of proteins from that messenger RNA strand. So the messenger RNA that was created in the nucleus will travel outside of the nucleus and will locate either to the cytoplasm or the rough endoplasmic reticulum, searching out ribosomes for the ribosome to translate the message and to actually create the protein. So graphically, you can see a picture of this. You see the nucleus is this big green circle. We have the DNA in there. We have the messenger RNA being created through transcription. The messenger RNA then goes outside of the nucleus, finds the ribosome, which is the blue, weirdly shaped object. The ribosome reads the messenger RNA and uses that information to create the polypeptide chains, or the proteins. And you can notice that these oddly shaped, almost T-shaped, are your transfer RNA, which are going to bring those amino acids to the ribosome so they can link and form the growing protein chain that you see in the middle. That big, long, green little circles all linked together. Translation is also a highly complex process like transcription that goes into way more detail than what I'm going to discuss with you this year. So only thing you need to know about the steps is what I am describing here today. So step 1, the messenger RNA has to leave the nucleus and search out and bind to a ribosome. The actual 5' end of the messenger RNA binds to the ribosome. Notice something else that is being made in the 5' to 3', or being read in the 5' to 3' direction. So you can see in the picture, the ribosome binds to the messenger RNA at the very edge of the messenger RNA. The ribosome will not instantly start translating the message. The first thing it has to do is look for a start codon, which is usually the nucleotides A, U, and G next to each other in a messenger RNA strand. So the question is, what is a codon? So a codon is a group of 3 nucleotides on a messenger RNA that specifies one amino acid. So it is 3 letters that in a row that will code for a specific amino acid. AUG is known as the start codon because it codes for starting the actual translation and protein synthesis. The question is, how do you know what amino acid each individual codon actually codes for? So they've created something known as a codon table or a codon chart that you will be responsible for knowing how to use. We'll practice with these in class. So you can see, let's do an example. If the codon reads, let me get my pen out. Let's say the codon is U, U, U. So the way you start is you work on the left-hand side, which says the first base in the codon, that's U. Then you go to the top, which is the second base in the codon, that's also U. And then you look for the third base in the codon on the right-hand side, that's also U. And then you look for where all 3 meet. So this row, this column, and the first one we see that UUU encodes for the amino acid phenylalanine, which is the abbreviation PHE. You do not need to memorize which abbreviation is which amino acid. You just need to know how to read the chart and then figure out what the amino acid's abbreviation is. One thing I also like to point out is you notice in the chart there are a lot of repeats. So there's 64 codons. However, remember there are only 20 amino acids. So therefore there have to be repeats. And this is actually known as the universal genetic code. Universal as in almost all living things on this planet use this genetic code. Some things, like your mitochondria, have slight differences in the codons and the amino acids, but everything else from a small bacteria up to you, up to an elephant, all have the exact same codon chart. So you can use that when we talk about bioengineering and how we can actually create human hormones like insulin inside bacteria using this fact. And we'll, as I said, we'll practice with this way more in the coming days. After the ribosome has reached the start codon, usually AUG, amino acids that are attached to tRNA molecules are then brought over to the messenger RNA. The tRNA at the bottom of it has an anticodon that is complementary to the codon on the messenger RNA. So you can see the picture at the bottom right. The anticodon is UAC. That will then base pair to a matching codon on the messenger RNA. So that's how you know that the correct amino acid is being put in the correct place, because the anticodon has to be complementary to the actual codon located on the messenger RNA. So the actual definition of an anticodon, anti means against, so it is the group of the 3 unpaired nucleotides on a tRNA strand, and they will bind to the messenger RNA codon using the base pairing rules. So you can see here's a picture of a tRNA. Notice it has that characteristic T shape. At the top, the AA is whatever amino acid, methionine, phenylalanine, serine, whatever it is. And at the bottom is the anticodon that will then match the codon on the messenger RNA. So let's say this is just a random example. This is not probably right. But let's say that this is actually methionine, met. Therefore, this codon will code for methionine through this anticodon base pairing. The tRNA anticodon will bind to the messenger RNA codon, and then we'll add an amino acid to the growing chain. Each amino acid matches up with 1 to 6 tRNA molecules. So as we discussed, there's 64 different codons, which means there are at least 64 different transfer RNA molecules. And as we've discussed, there are repeats, so there's multiple transfer RNAs that will have serine or have methionine on them. After the base pairing occurs and the amino acid is added to the growing chain, the tRNA will leave and go off in search for another amino acid to bind, and the amino acids that are left in the ribosome will then be joined together through a polypeptide bond. The ribosome will be continue to read the mRNA sequence until it reaches something known as a stop codon. Just like the start codon, there's a codon, a sequence that codes for stopping the translation process. At this point, the amino acids will disconnect from the messenger RNA sequence from the ribosome, and the almost complete protein is then released into the cytoplasm, where it then can fold properly, hopefully, and do its function. So you can see at the bottom, there's the picture of it. You see that the growing amino acid chain, the leucine, alanine, asparagine, and leucine. And you can see the next one is an incoming tRNA molecule that will add an alanine. And this will continue to be done until it reaches that stop sequence, that stop codon.